Hi, Miss Nikki here. Welcome to Chapter 16. This is Part 3 where we're going to discuss the ear. We have two kind of major topics to talk about: hearing and equilibrium or balance. So I'm going to go through both of these topics. We'll talk about the structures necessary to understand the physiology and then do kind of a short overview of what does it take to turn sound into an electrical message that the brain can understand, or how do we take balance or sensory information, generate an action potential, and again have it perceived by the brain. Ear detects sound and head movement. We're going to generate action potentials, and they're going to be transmitted to the brain via the vestibular cochlear nerve. Remember, this is cranial nerve number 8. So the vestibule has to do with balance or equilibrium, and the cochlea has to do with hearing. And then we can break the ear down into 3 sections. So we have the external ear, we have kind of this middle ear, and then we have an inner or internal ear. So external ear, we have structures. 1, the auricle. This is the part of your ear that you can touch and bend, right, because of this elastic cartilage that we have. And it's going to protect the earway, but it's also really going to direct the sound waves inward. So the sound is being directed through these sections of the ear because hearing really happens here at the inner ear. 2, we have the external acoustic meatus. This is your ear canal. This is where we're producing cerumen or earwax. You can also call it the external auditory meatus. Either term is fine. And then we have the tympanic membrane. This is your eardrum. So between the external and the middle here, we're going to have our tympanic membrane. It's kind of the separation point. Here's a better visual representation for you. Here's our ear that we're familiar with. This is the auricle. Then we have the external acoustic meatus. So the sound waves are entering or being funneled through the ear to the external acoustic meatus, and then you can see the tympanic membrane right here. And all of these structures make up the external ear. Now we're going to move into the middle ear, and you can see that the structures that are actually responsible for hearing and equilibrium are embedded in the inner ear. So one of the things you're going to see is sound waves. Let me change colors. Sound waves are going to be funneled from the ear to the external acoustic meatus, and it's going to cause the tympanic membrane to vibrate. And then these bones are going to move, and they're going to push on these structures, and there's fluid in the vestibule, the semicircular canals, and in the cochlea. And that fluid moving is what's actually going to generate the action potential that travels on the vestibular cochlear nerve. And if you can see, part of the nerve is attached to the vestibule, and part of the nerve is attached to the cochlea, and then it merges to form the vestibular cochlear nerve. In the middle ear, we have anything after tympanic membrane. So after tympanic membrane, you have tympanic cavity, and this is where we're going to have the ossicles, or the bones of the ear, that we'll see on the next slide. You're also going to have the auditory tube. Used to be called eustachian tube. We're really trying to stay away from eponyms, so it's the auditory tube. And the auditory tube is going to lead to the nasopharynx. It's usually closed. So this tube is usually narrowed or closed, but if you yawn, it allows air to move through. So this kind of equalizes pressure between the outer ear and the middle ear. So if you've ever been on a plane and all of a sudden it's hard to hear, and you hold your nose and you swallow and you hear a popping noise, you're really equalizing the pressure here in your auditory tube. The auditory ossicles, you need to know their names. The malleus is the first, the incus is the second, and the stapes is the third. The malleus looks like a hammer, the incus like an anvil, and the stapes like a stirrup. Here's the malleus, here's the incus, and here is the stapes. So what's going to happen is we're going to see sound waves enter the external acoustic meatus. It's going to cause the tympanic membrane to vibrate. That is going to cause these bones to vibrate. So they're kind of amplifying the sound. And the stapes is pressing on the area where we said we have fluid. And I'll show you more of this as we move further along. There are also 2 muscles And these muscles will contract if there's a really loud noise to try to dampen or stop the stapes from causing the fluid to move. So it's kind of like a safety measure. Finally, we get to the inner ear, and we're going to see a bony labyrinth inside the temporal bone. So you're going to have these kind of carved out spaces, and then we're going to see a membranous labyrinth. And this is going to have a membrane, it's going to be fluid filled, and we're going to see endolymph in the membranous labyrinth, and we're going to see perilymph in the bony labyrinth. Finally, we get to the organs that are responsible for hearing, and both of these are responsible for balance and equilibrium. I know I keep talking about the vestibule first because that's where we have the stapes and the oval window pushing on the vestibule. You'll see the semicircular are connected to the vestibule. And I promise this will all make sense. We just have to go through the structures first. We can put all this together in the inner ear if we look at the structures. So here we have the semicircular canals. At the base of the semicircular canal, they have ampullas. These ampulla and the semicircular canals are going to have something to do with rotational movement. So when I think of rotational movement, I'm really thinking of spinning. Then we have here the vestibule. And the vestibule has areas called the utricle and the saccule, and these are going to have to do more directional movement. And what do I mean by that? I'm talking about forwards and backwards, or up and down. And finally here we get to the cochlea. So you can see all of this is the cochlea. And I really just want you to pay attention to the fact that there is fluid inside these structures. So we have perilymph and we have endolymph, and this is going to be important. This is the fluid that we're moving around. So remember, the stapes is here pressing on and forcing the fluid to move within the inner ear. Here's a close-up of the cochlea. So we took a section of the cochlea, and now we're looking at just one of these sections. I want you to see here the fluid. So this is the perilymph, and this is perilymph, and this is the endolymph. And we can see here, this is the cochlear branch of the vestibulocochlear nerve, cranial nerve number 8. And you can see that nerve is coming to this section right here, where we have something called a tectorial membrane and a basilar membrane. So we're going to enlarge this section on the next slide, and we're going to talk about what's happening. This is called the spiral organ. So here is the cochlear branch of the cranial nerve number 8, vestibulocochlear nerve. And I want you to see we have the same pattern. Hopefully you're realizing with these special senses, it's always a cell with some sort of hairs, right? Those hairs are going to have something bind to them, or they're going to move, and that cell is going to generate an action potential which travels on a nerve. So what's happening is we have this tectorial membrane here, and the cells are attached to a basement membrane. Remember, below we have fluid that's moving. And what's happening above? We have fluid that's moving. So remember, in the other image, this was all perilymph. And down here was perilymph. So that fluid is moving and pressing on the bottom, the basilar membrane. So what's happening? If you have this tectorial membrane, which you can think of it like a ceiling, and you have this basement membrane, or basilar membrane, that's the attachment point. And remember, this is all fluid that's moving, right? So it's hard to imagine fluid moving beneath you and pressing you up, maybe if you're at the beach and you're kind of floating on the waves. I also like to think of this as like a bouncy house, except for with the bouncy house, it's air, right? So what happens if somebody jumps right in front of you? It's going to push you up, right? You're going to go flying in the bouncy house. So what's happening is the fluid is moving, And that's pressing on the cells here, these supporting cells of the spiral organ. And these cells have little hairs on top of them. And what happens when the hairs reach the ceiling? They bend. So if you have a bend in the cilia, that is enough to generate an action potential, and then that action potential will travel on the vestibular cochlear nerve to the brain. If we follow the pathway for hearing, how do we turn a sound wave into an action potential? So the sound waves are going to enter the ear, they're going to vibrate the tympanic membrane, which is going to cause the ossicles to vibrate, and then because the stapes is going to be at something called the oval window, that's pressing on the fluid that's inside, right? Remember the endolymph and the perilymph. So specific regions of the basilar membrane are going to move. Remember the basilar membrane with the cell and the hairs. The hair cells are going to be distorted. They're really going to be bent, right? They're being pushed over because what's at the top here is that tectorial membrane. This causes changes, causes neurotransmitters to be released, and that information is going to travel along cranial nerve number 8. If we start from the top, we can see the sound waves moving through the external acoustic meatus, causes the tympanic membrane to vibrate, causes the ossicles to vibrate. The ossicles are then pushing on the fluid, right? The endolymph and the perilymph. And those vibrations are pushing up on the basilar membrane. When the basilar membrane moves up, the hair cells are pushed into that ceiling or tectorial membrane. Their tips are tilted or distorted or bent. That's going to allow an ion channel to open. We're going to see depolarization of the hair cell, and that's going to cause the hair cell to release neurotransmitters, which can then excite the sensory nerve and send a message to the brain. So basically, we're taking sound waves and we're turning them into action potentials. So here's the visual representation. Here's the basilar membrane. Remember that the fluid is being pushed up. That's causing this hair cell to be pushed against the tectorial membrane or the ceiling. That's going to distort these stereocilia, and that's going to cause a potassium channel to open, and you're going to have depolarization. And what that's going to cause is neurotransmitters to be released from this cell that'll then be picked up by this sensory neuron, which is cranial nerve number 8. So you can see why we call these special senses by now, I hope. There's basically a cell that's being acted upon, and that's causing it to generate an action potential. Sound is the perception of pressure waves from the vibrating object. So the vibrating object is going to push air molecules, and this transfer of energy from one molecule to the next is what we're actually hearing. So if you have an object here, and this object is vibrating, it's pushing air molecules, and those air molecules that are being pushed, that's what's actually entering our external auditory meatus, and that's what we're measuring, these pressure waves from a vibrating object. There is something called pitch, which depends on the frequency of the vibrating object. Think about if somebody can hear something in high frequency or low frequency. Also think about here, humans can hear about 20,000 hertz. So just for comparison, dogs are about 45,000 hertz. Bats are at about 100,000 hertz. Something lower, like an elephant, they don't have really good hearing. Theirs is about 12,000. I did want you to see that certain sections of the ear are responsible for high frequency versus low frequency. So somebody could have damage in a certain section of the basilar membrane that would reduce their low frequency hearing, but maybe high frequency, everything's okay. They don't have any issues. Loudness depends on wave amplitude. So remember, we have our object vibrating, it's pushing or compressing air molecules. So you had air molecules all over. When this object is vibrating, it's pushing with force, with energy on these air molecules and causing them to consolidate. And then the next set gets consolidated. So this would translate into kind of this wave pattern. So we're really looking at loudness depending on wave amplification, which has to do with the compression of air molecules. I want you to realize that we can hear at about 0 decibels. This is if you hear, let's say, leaves moving, right? Leaves rustling. Anything over 85 decibels can do harm. And just to keep this kind of in perspective, a gunshot can be anywhere from 120 to 150 decibels depending on what type of gun is being fired. So this is, just one time not having earplugs in when you fire a gun, and you could be doing harm to your cochlea. And just to make sure that we can follow, we should know that the vestibular nerve and the cochlear nerve merge to form the cranial nerve number 8, vestibular cochlear. That nerve is going to enter here at the pons, and then the midbrain, travel to the thalamus, and then eventually that sensory message is going to make its way out to the primary auditory cortex, which is in the temporal lobe. Deafness is defined as any hearing loss, but there are 2 different types, whether we're talking about some transmission disruption between the external and the middle ear, or whether there's a malfunction at the inner ear itself. Equilibrium is the awareness and the monitoring of head position, and this is going to be done by the vestibule and the semicircular ducts. You can see the utricle and the saccule, if you remember these are in the vestibule, they're going to detect static equilibrium or linear acceleration. This is forwards, backwards, up, down. The semicircular ducts are going to detect angular acceleration, and this is where we talked about the ampulla. Another figure for us to look at, we know we have the vestibule and we have the semicircular ducts. We're going to talk about the utricle and the saccula, and specifically this structure called the macula. So this structure here and this structure here, the macula, is what we're going to look at on the next slide. I bet by now you could look at this image and you could tell me what's going to happen before I tell you. We have a cell. We have hairs. We have a nerve branch. So what do you think is going to happen? We're going to bend the hairs. They're going to cause the cell to depolarize, and that's going to generate an action potential that's going to travel on this nerve. The macula is the receptor. It's located in the utricle and the saccule of the vestibule. It's composed of hair cells and supporting cells. We're also going to have this gelatinous membrane, and inside of it we're going to have calcium carbonate crystals. So some people like to think of the otolithic membrane as jello, and the otoliths or rocks or fruit, for our example, in the jello. Here is the gelatinous membrane, and you can see embedded in the gelatinous membrane are these otoliths, these calcium carbonate crystals. We can see the hairs are standing straight up, the stereocilia. So what do you think happens if you move your head forward or backwards? What do you think happens to these hairs? The rocks are going to shift, the jelly is going to shift, and that's going to push the hairs to bend. Nice image here showing you that the otolith membrane, the jello membrane, along with the otoliths inside of the membrane are going to shift as you tilt your head forwards or backwards, and that's going to cause the stereocilia to bend in a specific pattern. If they bend back, that's a message to your head, my head is bent back. If they're bent forward, that sends a message to your cerebral cortex, I'm bending my head forward. So just as an overview, again, we can see that if the otolithic membrane along with the otoliths, if this shifts, Due to the change in your head position, it causes the stereocilia to bend. That's going to cause the cell to release a neurotransmitter. That's going to cause depolarization of the hair cell, and that's going to generate an action potential that travels on the nerve. And specifically, this nerve is the vestibular nerve, which will merge to form the vestibular cochlear nerve. Now we move on to angular acceleration. So the other one was static, forwards, backwards, up, down. This is angular or spinning. So there's a region called the ampulla, and we're going to look specifically at the cristae ampullaris. These are hair cells. I bet you can see what's going to happen. Oh, we got cilia again, right? So let's look at the image. We can see here at the base of the semicircular ducts, we have this ampulla. Specifically, this section is called the cupola, and this is again a jelly-like substance very similar to the otolith membrane. We can see the stereocilia here, we can see the hair cells, and we can see the nerve. So what do you think is going to happen? This is all fluid. This is endolymph. So as you're spinning, the fluid starts moving, right? So we have movement in the fluid that causes the cupola to bend, the gelatinous material. That triggers the stereocilia to bend. That's going to cause the cell to depolarize, right? Open up ion channel, depolarize, release a neurotransmitter, and that's going to generate an action potential in this vestibular nerve, which then sends sensory signals to the brain. If we wanted to follow the pathway to the brain, we know we're here at the cristae ampullaris. Then we have the vestibular nerve. It merges with the vestibular cochlear nerve. It's going to make its way to the brainstem, but it's also going to send messages to the cerebellum, because remember, this is about movement. This is where we collect information like proprioceptors, right? So the cerebellum is collecting information about your position in your place and time, right? The information is then going to travel up the brainstem to the thalamus and then eventually to the cerebral cortex, where you'll be aware of the spinning. So something that I want to mention here lastly, and then we'll move on just because I think it's interesting. So why is it that people get seasick? Why do you get nauseous? So we've talked about vision already, but let's say your vision, you're in a cabin, you're on a boat, but you're in a cabin. Your vision is telling you you're in a room and the room is stationary. But your ear is telling you we're moving, right? Movement. We have movement. Your brain is trying to process. Your brain is saying, okay, somebody's lying, or we're on drugs, or something is wrong because my vision and my equilibrium are not matching up. And so what can I do if you just ate a plant or you ate something? Evolutionary wise, it makes sense. If your brain thinks you're hallucinating, I'm going to make you throw up, right? You probably ate something that's bad. So we think that this is why some of us have that reflex. I can't read in a car, it makes me ill. I'm trying to focus on the page and be stationary, but my inner ear is moving, no good. Also, something called vertigo. Sometimes you can have the otoliths from the vestibule make their way to your semicircular canal. And if these calcium carbonate rocks are in your semicircular ducts or canals, it makes you think you're moving even if you're not, because those rocks are making the fluid move. And that's causing the cristae ampullaris to move. And so you think that you're moving or spinning, but you're really not. It's just the otoliths somehow dislodged from the otolith membrane, and they're kind of wreaking havoc on your angular sensory messages, if that makes sense. So we can give you medication to kind of quiet this nerve transduction from your vestibule and from your semicircular canals to kind of tell your brain, okay, don't listen to those messages. All right, well, we are finally finished. I hope you enjoyed this special senses topic. I think it's really fascinating, and you can see why we call these special senses. We're always having a hair move, and that hair is causing the cell to depolarize, and that's causing a neurotransmitter to be released, and that's causing the message to be sent to the brain. So hopefully you had fun, and I'll talk to you later. Bye.